my name is Suzanne. I'm an application scientist for Nexcelon Bioscience, and today I'm going to be doing a video demonstration of the newest instrument that we have. It's the Cellaca MX. The Cellaca is a high throughput cell counting and viability testing instrument, and how it works is it will count 24 samples in one batch. You can use trypan blue or you can use a fluorescent viability dye. We suggest you use acridine orange and pipridium iodide, or AOPI. And what I'm going to show you today is a demonstration of how quickly and accurately we can count 24 samples using trypan blue and AOPI. The Cellaca requires you to pipette your samples into quite an ordinary looking 96 well plate. In fact, these are custom made. What you do is you pipette your samples into row C and row G. So there's 12 across there, 12 across there. And the imaging will take place in rows B and rows F. We have rows H and rows D that are mixing wells, so you can mix your samples with trypan blue or with AOPI in those wells. And then once you are finished pipetting your samples, you can then place your plate into the Cellaca. So as you can see, the software looks pretty straightforward. We've got a number of tabs down the left hand side, and you work your way down those tabs. So at the top we have the load plate tab, which will automatically come up when you load the software. After you've loaded your plate, you'll then move to setup, preview, counting, and then get to your results as well. So we also have an automation tab here. So we have lots of these instruments that are connected to automation systems. And if that is the case with your instrument, that tab will also be available. So when your software loads up, you'll automatically be on the load plate screen, and underneath the eject plate and load plate button is where we need to give the plate a name. I'm going to call it cell test with trypan blue. And the next thing to do is to click setup. So now we select the trypan blue default assay from the drop-down list, and the next thing to do is to make sure that the correct wells are selected. If they're highlighted in blue, they are selected and they can be clicked on and off with your mouse. So you can then move to the preview tab and preview your cells with this button. So now I'm on the preview screen. I can preview one of my wells. You can see the cells in there. You can change the focus if needed, but this looks pretty good to me. So once you're happy with it, you can stop the preview and press the count button. So using trypan blue and using the Cellaca like this is really useful for people who really need to save time in their cell counting. Because we're just taking an image in the brightfield channel here, there's no red and there's no green. It means the Cellaca can perform those 24 viability tests in a super quick time. So you can see how quickly those wells are now turning from blue to yellow to orange and then to green, meaning everything's been acquired, counted and saved. It all does that really quickly, and in fact the 24 samples are completed in a total of 48 seconds. So that's just 2 seconds per sample, which is great for people needing to do multiple cell counts with all of their samples. So that is all 24 samples counted in a total of 48 seconds. And now let's take a look at the results. So on the results page, the viability is pretty comparable all the way down, where we're almost all the time we're within that kind of 90 to 95% viability, which is great. But also let's have a look at some of the images as well. So here we can see the here we can see the cells. With all the live cells, you will get a green ring drawn around them, and the dead cells will have a red ring drawn around them again. So again, we can see really nice segmentation. Whenever there are cells close together, they're segmented and counted correctly. And you can see that all of these dim trypan blue positive cells, so dead cells, are being picked up really accurately as well. Now we can look at an alternative method of viability testing, and that requires using a fluorescent live dead dye. It is a combination of acridine orange and propyridium iodide, or AOPI, and what it will do is it will turn your live cells green and your dead cells red. So you simply mix the dye, the pre-combined dye, with your cells 1-to-1. There's no incubation time, there's no extra reagents needed. After you've mixed it, you're ready to load your Cellaca plate. Okay, so once your plate is loaded into the Cellaca, what we can do is we can now look at the software. And the first thing I'm going to do is give my plate a name. So I'm going to call this Cell AOPI Test 1. Once I've given the plate a name, I'm going to tell it about the setup. Just like our cellometers, you can have a mix of different cell types, a mix of different assays, all within a drop-down menu here. So anything that you're currently working on in the lab, you can have an application for. I am just using the default settings for AOPI, so a fluorescent viability assay, and I have a sample in every single well. So I have all of my wells selected. You don't need to use every single well. For instance, if you have an experiment where you only need to test 6 samples, we can just test the first 6 or the first 12. But with this example, I want it to scan in every well. So next I want to preview my sample to make sure everything's set up correctly. So I'm going to go to the preview page. We can preview any of the wells, but let's stick with A1. We can see the cells in brightfield here. What we can do now is we can focus using these focus buttons here. And what I'm going to try and do is make sure the cells are nice and in focus, like so. What I also want to do is preview the green channel, which is the FL1 channel. So I want to make sure we've got some nice live cells that you can see there. And also preview the red channel as well. And we've got a couple of dead ones as well. Once you're happy with the exposure and the focus, you can stop the previews and press the count button. So what the instrument is going to do now is it's going to take a very large image in each of the wells. And what it will do is it will add up all the live cells and all the dead cells and give you a concentration as well as viability for each and every well. So you can have a different sample in each well if you want to. You can have replicas, you can have them in sets of 3, sets of 4. You can set the experiment up however you like. But the most important thing is it's going to do this extremely quickly. So you can see that the wells, A1, A2, A3, they're currently going from a blue colour to a green colour. When they go to a green colour, that means the images have been taken, the counts have been done, and the data has been saved. In fact, they'll go orange first, which means the counting's in process. Then they'll turn, green, which means everything's been saved. Doing an experiment like this with AOPI, you can count 24 samples in around 2.5 minutes. So that is going to be way faster than any other method out there. It's also, you don't have to wait here. You can go and you can do other things as well and leave it to count for those 2.5 minutes. But hopefully we should see some nice results at the end of this. So while the rest of that plate is running, I wanted to explain how the live dead AOPI fluorescent dye works. So AO, or acridine orange, is a green dye, and if you were to use that on its own, it would enter all live and all dead cells and everything would fluoresce green. However, we're using it here in combination with propyridium iodide, and propyridium iodide is a red dye. That only enters cells with a compromised membrane, As it enters, it will quench any of the green fluorescence that's in there and turn the cells red. The best thing about this combination of dyes is they are both nuclear dyes. They're not going to bind to anything without a nucleus or without DNA. So any debris or anything without a nucleus within your sample is not going to fluoresce with either colour. I also wanted to talk about accuracy. So the Cellaca will accurately count cells at a concentration between 1 × 10 to the 5 per mil and 2 × 10 to the 7 per mil. So if your cells are more concentrated, then a pre-dilution step may be needed. And finally, the Cellaca comes in 3 formats. It comes as a brightfield only version. It comes as a red-green brightfield version, which is perfect if you're just doing AOPI viability. And finally, it comes at the highest spec. It comes with a choice of 4 excitation and 5 emission channels, which can be used in different combinations and is perfect if you are using alternative fluorophores to what we are showing here. So once the Cellaca has finished counting, the results page will automatically pop up at the end, and this was the same sample that I pipetted into every well, so I'm looking at reproducibility here. So I'm looking at their viability results that are down here that you can see with my mouse. They all look really similar, which is great. I'm going to check all the way down to the bottom. They're all between 91 and 94, it looks like. I'm also looking at the concentration similarities down the plate as well. It looks like here like it's between 1 × 10 to the 6 and 1.3 × 10 to the 6. Again, across 24 samples. That's pretty good reproducibility there. But we don't just expect you to believe the numbers that is given to you. Let's have a look at the images as well. So if we click on the images, you can see your combined images here. So this will be in green and in red. And when a cell has been counted, a little green circle will be drawn around it if it's a live cell, or a red circle if it's a dead cell. This is a pretty viable sample, so we can see lots of green cells there. I'll see if we can find some red ones. Yeah, here we go. We've got some dead cells here that have been circled in red. You can toggle that counting on and off just to make sure that everything is being counted correctly. These are the default settings, which are normally really good. You don't need to change much, but if you do, the settings are optimizable as well. I want to try and find, try and look in a different well, just a well across the plate, just to see what we can see there. The cells are laid out slightly differently here, but let's have a look. We've got some more dead cells, and we've got some, here we go, we've got some nice clusters of cells here, 2 little clusters at the bottom. If I uncheck that counted button there, we can see there are 3 live cells here, 3 live cells here, 2 live cells, and a dead cell here, all really close together. If we add the counted option on there, you can see that the declustering is really, really good. It's going to give you the most accurate results in terms of cell counting, especially for clumpy samples. So the last thing I want to show you is how everything gets saved. Everything gets automatically saved. You don't need to remember to press a save button at the end. I'm just going to open the file that everything has gone to, what I've shown you today, and you can see that the 2 files are there. We've got the AOPI test and the trypan blue test. Within those files, you have 24 different folders. Those contain images from each of your wells. So for each of your samples, you have a set of images. For trypan blue, you just have a raw image and a counted image that you can look at there. If we go to the AOPI option and look at the same thing again, in this one you'll also have the fluorescent images as well as the count data. In the root of those files at the bottom, you will also have all your cell type settings, your assay type settings, just in case you ever need to come back to them and to look at them again. They're all automatically saved there. You also get a data file that can be opened in Excel, and that will give you all the data from every single sample within that plate that you've tested. So it's all in one nice neat file. And the final thing at the bottom here that says manifest, if you open that within the software, you can reopen all of your images. You can change the analysis settings. Say you wanted to do a different type of analysis on them. Say you use the wrong settings by accident. You can open all those images again and analyse them completely differently if you want to. So that option is always going to be available to you, and those checking options are always available as well. So I think that's everything. I have put a link to our website where you can learn a bit more about the Cellaca, and also put some email addresses up there where you can contact us if you would like to hear a bit more, or if you would like a personalised live virtual demo of the instrument, we can do that, or you can also get it in your lab to test it out as well. I hope you enjoyed it. Thank you for listening.